Hi and welcome to Nervous System Part 2. Second part here, we're going to be talking about the electrical signal, how it travels, what the difference is between a graded and an action potential, and then synaptic transmission. I will split those up into smaller lectures. Up to now, we talked about the resting potential, and now we're going to move on. So when we think about a neuron that's actually depolarizing, the question is, does it do it all at once? So if you look at the neuron again, and we had the cell body and the axon, and it's going to travel down that way, what's the sequence of events that happens, and how does that occur? So in this slide, you can see that there's the input zone. We know that sensory is coming in through the dendrites. It's arriving at the cell body, and from there, in the trigger zone or the axon hillock, remember that term, it's going to generate an action potential that travels down to the end zone and the conducting zone. So we want to look at that flow of current more closely. We're thinking of that current or potential, which is the flow of charge, which are those electrons that are moving. So we call that, as I've been calling it, action potential. And I've written out the definition for you there. So you have a really nice definition, and that is the momentary change in the membrane potential that serves as a long-distance electrical signal. Remember, there's going to be a change in the membrane permeability to sodium, correct? So that's what I mean when I say a change in the membrane potential serves as a signal. So let's look at it now closely in pieces. First, here's the axon, and on the axon, the axon hillock here is going to be the trigger zone, right? If that intensity of that message is sufficient, then it will reach the axon hillock and this neuron will fire. Okay, when I talk about intensity, I mean you don't feel everything. You don't feel a mosquito that lands on you sometimes. Now, there is a signal there coming in to some of these neurons, but it's very faint. And it's so faint that, let's say, it doesn't actually, it comes in through one dendrite or two, and it doesn't really trigger an action potential. So that potential needs to be, we call it a threshold, above threshold. So you see here, let me change to pen. Here is written for you, above threshold. If it's above threshold, then it's going to fire and it's going to go all the way down to the end of this neuron. It will never peter out and not quite get there. So once we've triggered it at the axon hillock, we're calling this then the active area at that peak of that action potential. And what happens is that a sodium channel is going to open there. And sodium is going to come in, and inside the cell is going to now be, just in this very first section, positive in relationship to the outside. Whereas the rest of the cell here is still negative in relationship to the outside that's positive. So we're starting here at the region of the axon hillock. Sodium is going to come in, and there's this change in that charge inside the cell. That change is going to trigger the neighboring section here, because those ions attract each other, trigger that neighboring section to be stimulated. And if it's stimulated, what's going to happen? The sodium channel is going to open there. And then it'll stimulate the next one. And then it'll stimulate the next one. And the next one, as this action potential or charge change goes stimulating the neighboring area and traveling all the way down to the end of the axon. Okay? Here in blue, it's telling you the adjacent inactive area into which depolarization is spreading. It'll soon reach threshold. So it's going to happen because I've already changed the charge in this area here. So switch, 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 and we'll travel every single place here. There's a sodium channel and a sodium channel and there's sodium channels all the way along here that are going to open, letting sodium in and turning this very positive in relationship to the outside that would be negative. And that's the direction of the propagation of the action potential. It will always reach the other end, remember? And it'll then, connect to another cell in the location where it touches any other cell. So that's how it's flowing. If we looked at it, if we zoomed in, this is now the depolarized section that's moving down this way, correct? And it's going to turn this one into positive, and then moving on to the next one, and it's going to turn this into positive, and it's going to be traveling down. So that's understood, but we also want to know what happens to the region that was left behind. If this is the active region, then what about the one that you already went through? Well, slowly that is going to, I'm going to just write a big R here, that's going to repolarize. So this will be repolarizing behind it. So there's a slight lag time by which this area cannot be re-stimulated until the next one comes. How much time? Well, remember that this is all happening. It'll get all the way down an axon in as little as milliseconds. 2 milliseconds. So this is incredibly fast. And I'll give you numbers later on for what's happening behind it when we look at how it's blocked a little bit. So that blocking it immediately behind the active area allows it to not go both ways. Because if we want it to move this way, if, however, this positive could potentially stimulate what came behind, then it would move that way too, right? And this does not happen. There's enough of a lag time to reset these channels here, not allowing that action potential to move in the wrong direction. What opens the channels? Well, remember, it could be voltage gated. Remember, we're illustrating it as a little ball. It's not really a ball. Channel could be closed. It will be stimulated, for example, by voltage. In this example, it's stimulated by voltage because it's the voltage of the previous area that is stimulating the channels that are next to it to open. If we made a trace of an action potential, what would it look like? Remember, I drew this for you, but now we're going to look at it in a whole lot more detail. We're going to take all these pieces. Remember that the way we did this is we get a neuron and we will hook it up so that we have a recording electrode here and then a second one outside. I'm going to make another one here, outside the cell. And so you're getting the difference, potential difference between inside and outside of that cell. And we want to get a trace. If you were to record that during a stimulus, during an action potential, where we know it's turning positive, then we're going to have this curve that goes from being very negative in the -70 to being much more positive, 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 overshooting the 0, overshooting that, coming all the way up to about +30 before it starts to gain potassium, lose potassium ions and come all the way back down. But really I want to take you through those one step at a time in the next video. So our next video will start out with step 1 of this process of the action potential in much more detail. Thanks.